I wanted to move this up because what I'm talking about follows perfectly from what Warren was talking about. And I think the discussions that we were just having about data sharing and issues of consent and privacy and identification are all the ones that we've been thinking about a lot as well. So I wanted to kind of jump ahead and share what we've been doing and we've been thinking about. I should warn you at the beginning that this talk doesn't have an end. We don't actually know yet if we're going to be able to succeed in what we're talking about. But I will tell you exactly what's going on now because I think it's pretty exciting. So I'm going to talk about two very different entities but related. And these are the so-called Global Alliance and an effort that we've started at BRIITE referred to as Genome Bridge. And I will introduce both of these as we go along. To begin with, though, following on to what Bob had said about big data, this is a quote from the Big Data to Knowledge Centers of Excellence, RFA, which I actually quite like. And this argues that research is getting difficult because issues of accessing data, issues of working with data, all of the things that we should be able to address are getting in the way now at scale. And I think that this is the right thing to be thinking about. That we have for a long time in technology been doing what we can to support research, but often in a reactive way. And what I find is that a lot of what happens in research is tailored to the infrastructures that we build, right? So we constrain research by the infrastructures that we put in place, and then the researchers will adapt to how they use them. To lead this off, though, because Warren was talking about TCGA, I wanted to throw this in because this is where we're going with large-scale data analysis. This is the result of a paper now in press, and that analyzing the full TCGA data set looking for driver mutations. So this is now about not quite 5,000 tumor normal, matched tumor normal exomes. And the impressive thing is that in this one analysis, which took in our infrastructure a couple of weeks to run, the group was basically able to recapitulate about 20 years of cancer research. They rediscovered basically every known oncogene, as well as about 10% putative additional genes in the 21 tumor types that they were looking at. I mean, this is enormous, right? This is the value of what can be done at large scale with these kind of data. And there are a lot of genes left to be discovered. So if you look, this is showing frequency of somatic mutations versus discovery rate, effectively. And so this red line is relatively common somatic mutations. And these seem to be leveling off, right? These are the known oncogenes that we've been working with for a long time. But as you get rarer and rarer mutations, there's a lot out there that we just don't know. And we don't know it because we haven't had the power, the statistical power, to find these things yet. And the statistical power required is a function of how frequent mutations are in a given tumor type, right? So if you look at something like the environmentally driven cancers like melanoma and lung cancer, there's just a huge range of somatic mutations that come in. And you need a fair bit of power to resolve those, a little less down here. But the point is to really address what TCGA is trying to do, to build out this map. We're looking at hundreds of thousands of samples just to get to the point where we can reliably say we've cataloged the bulk of the mutations. And this is one of the lessons that comes out of genomics in general, and this is even more true for germline studies, is that we need lots and lots of genomes to understand any one given genome. And the challenge of amassing these many, many genomes is one that we've been thinking about for a long time. So NIH is probably funding something like 100,000 genomes this year, exomes, but in lots of little different places all over the place, right? So in groups of 100 or 1,000 or 10,000 maybe. But how do we leverage all of that investment, right? And at the same time, as head of IT, I'm looking at our computing infrastructure, which probably doesn't look dissimilar to your computing infrastructure, at least for genomics, with clusters of Linux machines and shared file systems behind them. I built my first cluster for genomics, I figured out just almost 15 years ago. And the fact that this architecture has lasted as long as it has is actually pretty impressive. But what we found in this TCGA analysis that I just showed you is that this infrastructure is creaking. This is starting to really, show its age. And we can throw a lot more of any of these things. I've got about 16 petabytes of disk spinning, and I can throw any more, more thousands of cores there. But the architecture itself is starting to get in the way. And this is something that I fundamentally believe we need to be thinking about now. We did an effort when I first took over as CIO about a year ago. We started an effort to go and interview the research community about how well research IT was meeting their needs. And a lot of the themes that come back were exactly those that were acknowledged in the BD2K RFA, that it's hard to find data, it's hard to work with data. I found this data, but I don't know what it means. I don't know how to find these tools. But one of the conclusions that we came out of is that our pretty substantial, I'm not displeased with the infrastructure we have, but it's actually starting to get in the way of what we can do with research because of the data we have. And so this is for the last, for a couple of years actually, there have been a number of us at BRIITE thinking about what does this mean? We have all of this data that we want to get together. There's data all around the world that we want to get together.
You mentioned the size of the infrastructure that's limiting. You mentioned 16 petabytes. What's your compute capacity there and network capacity, and it's still limited?
We're at about, 8,000 cores. We have a 10 gigabit infrastructure. What starts to become limiting, and I'd be delighted to discuss this with people because maybe I'm just getting it wrong, but what's become limiting for us is the file system as a data store. And this is something that we in bioinformatics have been leveraging forever, right? We leverage the file system as a data store. And the difficulty is, first of all, is a user interface model of having files and folders. That sort of breaks down we have about 2 billion objects, and that's just not, that's not an effective interface to get to those data. And that's one of those other things that happens at scale. There's a need to move from search to something that's more guided, that's more indexed, that helps you actually locate the data. And that requires effort that we haven't been putting in so far. So there are a number of these things that are coming up at scale that we just haven't had to think about yet. But the file system itself, it turns out to be really hard to build a Posix file system that scales out really well. So as I said, we've got about 16 petabytes of data. A lot of this is on Isilon storage clusters. We don't run ours bigger than about 3 petabytes for various performance reasons. And that's about it. It's hard to make a big file system that adheres to Posix compliance. And yet, in our field, we don't need it. Most of our data are immutable once written. We write them. We may read them several times. We may read them never. I think there's a fair amount of our data that is that write once, read never class of data. But it is, the file system itself is what is slowing us down. So we just did a large study looking at about 25,000 exomes. This is a germline analysis study using the GATK toolkit, which we developed, right? And the GATK toolkit, underneath the hood, opens up files, 1 file per sample or per chromosome per sample. And this means that at the Linux kernel level, you're having to manage something like 100,000 file handles. And that's not what Linux does well. And that's not going to get you to the 1 million file limit, right? So files aren't the answer, but files are what we have now. So this is the first piece of the problem. But the other is looking around at all of these other exciting big data opportunities and everything that Google does, and how do we make the Google for biomedical data? And I'm sure many, many of us have been thinking about that. But we've been focusing primarily on genomic data since that's what we make a lot of. And how can we leverage genomic data that's being generated not just in our institute, but across the country and around the world? And recognizing that the power in genomic data really only comes when analyzed collectively. And the potential to transform medicine, which is the ultimate goal of my institution and probably most of the institutions here, really requires that we think about it at this scale. So the scale is, we need to bring together very large reference data sets, if you will, of genomic data. And large on the scale of millions of genomes. And millions of genomes puts us into that space that our IT architecture doesn't play with very well. So even ignoring, for the time being, issues of permissions and data sharing and all of this, that's something at that scale that we're already stressing our architecture with. We need different software architectures to work at this scale. This GATK infrastructure that we've built that's being used broadly around the world for genomic analysis, we're already hitting the scaling limits of that. I mean, just fundamentally. We need new analysis statistical methods at this scale. So this is all of the big data stuff that's come in. I mean, that's been coming up over these last years. And the fear is that genomic data is racing towards the clinic. So we have places, we have a clinical sequencing facility now at Broad. There are efforts all around the community. One of our partner institutions, the Dana-Farber, will sequence every patient that walks through the door. There's a lot of this data out there. The fear is that these data, if we don't think about it now, end up in something that looks like an electronic medical record system, which I think we can agree from a research standpoint has just been a disaster. I mean, it's not an effective means of collecting and sharing data for research purposes. So this is really what has been our motivation.
I might make a comment there that not only is it not good from a research standpoint, from a decision support standpoint, it's been so incredibly hard to use EHRs to do decision support for clinicians. And that's, thinking about genomic data, that's what we want to be able to do. Right? It's decision support.
Yep.
These systems just don't do it well. So that's a whole other part of it.
We've done some decisions, with Mary Rowland at St. Jude and Delevin's.
But you do it outside of the EHR, actually.
No, actually, we've incorporated some decision support in the EHR in Millie, Cerner's product, but it's taken thousands and thousands and thousands of hours to get that infrastructure in place to report on maybe 50 polymorphisms in genes. So
It's not scalable.
It's not scalable, absolutely not.
So we've been talking about this at Broad literally for a couple of years about how can we build something that would benefit the community, a place where data could be stored, where people could come and access the data. What would this look like from a business standpoint? How do we make this sustainable if it's not going to be funded by research? And we talked, and we talked, and we talked, and we talked, and for a while it was going to be a commercial startup that we were going to spin out, and then we decided it had to be a nonprofit. There couldn't be a commercial motive in doing this, and we talked, and we talked, and we talked until a week after the last BRIITE meeting. So it was exactly a year ago, during the last BRIITE meeting, that one of the people in the extended Broad community reached into his relatively deep pockets and said, just show me that you can actually do something. And we went off and spent last fall prototyping in the Amazon cloud our high-throughput genome sequence analysis pipeline, so the GATK-based pipeline. We did it in a very brittle, naive way because we don't know how to think in this way yet, but this was an effort to prove, first to us internally, that we can do it, and then to go and start to sell externally the idea that the way we attack this problem is that we all work together, all now talking the global genomics community, and we do this leveraging some scalable, elastic architecture, not necessarily public cloud, although this is what we were working in, obviously, public commercial cloud. And so these were, once we got through this effort last fall, and in December of last year, we were able to show that we could run some relatively large analysis in Amazon, and we didn't tell anyone how much it cost and that sort of thing, because it's hard to do that stuff right. But this led then to a series of meetings, starting off with a very interesting meeting in New York in January of this year, of a number of people interested in genomics, either from a research perspective or a funding perspective or a medical perspective, to come together and create an organization to try to make this happen. And this is the Global Alliance. It is an awful name. I had nothing to do with the name. But the idea was to create an organization that functions somewhat like the W3C, that we need a standards body, if you will, to help promulgate ideas of data exchange, models of security, ways of dealing with consent, all of the issues that we're each wrestling with individually. We wanted to put together an organization to try to do this. So we got to talk to a bunch of people. We got people interested. We published a white paper in the early summer last year, and at which point we got 107 organizations from around the world. The number's bigger now. This is an old slide, but 107 organizations from around the world to sign a letter of intent saying, yes, we think it's a good idea to share genomic data. I mean, this is huge. And this represents some big funders. You'll note the Wellcome Trust is on here. NCI is on here, so Harold Varmus has been an active supporter of this, and you can see this, I think, reflected in the upcoming cloud RFAs that Warren mentioned, that to get to scale, we have to do this. There are a lot of countries here. And the issues of consent and data sharing and privacy are hard enough in one country. They're hard enough in one state. I mean, look at a state like New York. But to try to solve this across this morass is going to be, this is a daunting challenge. But this was exciting. It got ground right away. This was an article that was written in Nature Biotech that we had nothing to do with. There was no one involved with any of the 107 organizations or the steering committee for the Global Alliance, but recognizing that the community wants this. The community really wants to figure out how to do this. So it sort of falls to us in some ways to help support this. The Global Alliance is set up in 5 different working groups. The ones that are of direct relevance to us and IT now are these 2 around genomic data. How do we deal with genomic data at this 1 million genome scale? And what does security look like? And security is sort of a catch-all term there. So this includes how do we manage data use restrictions that come from patient consent, which in itself is a tremendously fun problem, it turns out. The Global Alliance today has an interim executive director. It has initial chairs for these 5 working groups. I've listed some of them there, people that you probably know. And the goal is that over the course of next year, that this becomes a real entity that's doing something useful. The white paper that we put out that argued the need for this anticipated that underneath this standards organization, there would be an open information platform. So if the Global Alliance is W3C, the open information platform might be, I don't know, HTTP probably, but something really very fundamental. And the paper further suggested that there be one or more so-called operating entities, which are the organizations that are actually building and operating this open information platform collectively. And so we started such an effort to build an operating entity under the auspices of the Global Alliance. And this effort is called Genome Bridge. This was formally established in May or June of this year, currently as a wholly owned LLC of Broad, but with the expectation that once we actually get the technology and funding model right and spin this out, that this will be an independent not-for-profit with its own board. And the focus here is entirely technical, and it's technical in all of the areas that I would argue that at least our research computing organization is not good at. These are the things we don't know how to do. We're not good at this fine-grained access control. We're not good at thinking in a large-scale distributed space. We leverage files rather than services, service APIs for exchanging data. All of these things that represent something very fundamentally different than we've been doing in the organization. Pulled together an interesting team. So Brad Margus, in addition to being a very experienced biotech guy, has 2 children with a very rare disease, awful disease, ataxia, I can't never pronounce it, thank you. Telangiectasia, ataxia telangiectasia. And has founded a patient advocacy group and is really coming at this from the patient advocacy perspective, that it's what we're trying to do is needed by the patients themselves. Eric Golan is an accomplished technical architect. We stole him from Carbonite earlier this summer. And then we have a chief medical officer who's one of these unusual people at Broad. He is an MD, PhD. While he was finishing his fellowship in cardiology and doing some additional work in machine learning, he's also working 25% time for Google Ventures and is really quite connected to the tech world. And he comes in as the chief medical officer. So we have this now in place, and we start to build on a vision. This is a very confusing, ugly picture, but the basic idea is we need some data storage and service capabilities, ideally not based on files, that can share both read-level data and variant-level data. We need some standardized best practice upstream analysis pipelines so that we can harmonize data that are being produced in different places so that we can actually begin to look at it together. One of the things that we find in genomics, whenever we work in a large multi-institutional project, if the sequencing is done at different centers, we all send each other our data and we all reprocess all the data from scratch because we can't compare data processed at our organization with data processed at another organization. So somehow we need to be able to create a standardized way for this to come together. And then the vision is to create kind of a third-party ecosystem that sits on top of this platform. Some commercial tools, some nonprofit tools that come in to leverage data here, potentially for clinical interpretation or for other forms of research. And so what has to happen underneath is that we think about a cloud architecture. And again, I would argue that this is something that at least our bioinformaticists don't know how to do. This is not what we've been doing for the last, since the Human Genome Project. This is not a series of Perl scripts that are calling Grid Engine or LSF. And I mean, Perl built the human genome. I like it, but It just doesn't get us to the next stage. This then directly impacts, so now wearing my Broad CIO hat, this directly impacts my future, the future of my service offerings in my organization. And so the goals here, I mean, just look at the two top-level goals, is we really want to take our big production pipelines and move them into this standardized operating entity. And we will commit to moving all of the genomic data that we generate into this infrastructure with privacy concerns addressed and data use concerns, all of those things addressed. This top line represents about 60% of the computation in my infrastructure today. So it's a pretty substantial impact to how we think we're going to move down the road. This second piece represents probably about 10 of the 16 petabytes of data we have. And I have to say, I am happy to get out of the storage business. I don't mind compute. You know, I've always been sort of an HPC guy. I can't wait to get out of the storage business. So, bringing us forward to where we are now. We decided that we needed a major scientific project to drive the ongoing development of this. And so starting about a month ago, it was actually not long after the ICGC meeting, actually, we came back and set out a goal to get all of the cancer data we can get our hands on, TCGA, ICGC, bunch of data that we have at Broad, miscellaneous data in dbGaP, and bring these all together to extend that first analysis that I showed you, which was done with about 5,000 genomes, to extend that 3- or 4-fold in terms of power. There's two goals here. So there's the scientific goal that we want to, I mean, this is going to be an awesome analysis if it works and we can pull it off. But there's also an engineering goal that through this process, we really do build something that supports this scale and is usable by more than just this effort. We decided just last week to do in parallel a large germline study, so initially 25,000 exomes, potentially by February 80,000 exomes. So we're really sprinting, trying to build this thing and make it work. This effort, by the way, has an awful name. So Firehose is our internal cancer genomics pipeline. Genome Bridge is this thing that's going to build it. FireBridge is the name of this big project that we're working on, because we like this better than Genome Hose, although that's kind of what we refer to it internally sometimes. And this is now a very focused effort. So this is all of our cancer genomics development team and the growing development team of Genome Bridge and some of the sequencing pipeline development team, all collaborating together, trying to build something that's going to solve these problems. At the same time, there are any number of operational and organizational and business issues that we haven't addressed at all, although I'm happy to talk about things like that. But issues of IP and issues of, you know, what's the business relationship between Broad and Genome Bridge and all of these sorts of things. But the idea is that there's going to be an awesome scientific goal coming out of this, very aggressively. I didn't want to write the time scale here, but I think we have announced it. It was announced at NCI a week or two ago. That we're going to start to learn ourselves how to use this, so that our researchers figure out how to use this, and that this forms the model for our next generation infrastructure. And then finally, that we actually give Genome Bridge some standing moving forward. So.
That was a closed meeting at the NCI, by the way. So that wasn't an announcement, unless there was another meeting that was open.
Yeah, well, I didn't want to do it here. I mean, yeah, but once it's announced there, it's everywhere. So, I mean, I've heard it all over the place, but I didn't want to write it down here. That's for other reasons too. And I think one of the other reasons I wanted to change the order of the talks is because I think the disaster talk following this may actually be relevant to this. We'll have to see. So this is kind of where we are today, right? We have the beginnings of something. It's not well connected to much of anything yet. There are, as we work through this FireBridge process, we are beginning to deal with, you won't be surprised, the challenges of permissioning all the data that we want to use. I mean, there are these processes that are just very cumbersome and slow when you're trying to do them at scale. And this comes back to some of the other things that you were talking about, needing to really change how data are consented for use in research. Because the process of having to go sample at a time back to a dozen different IRBs to, yeah, and it absolutely isn't scalable. Yeah, happily it's not, well, it feels that bad. I mean, really, they've been dribbling in as we've been doing this. This is partly why we've decided to do this germline process in parallel, because we already have consents on those, or we have permissions on those data, the right to use on those data. We are today piloting now in Google. So the Genome Bridge has a formal mission of being cloud agnostic. So we did our first pilot at Amazon. We're working in Google now. We're actually working pretty closely with some of the Google engineers who are starting to get excited about this project, which means it's big enough to be interesting. I mean, you have to be relatively big to get Google excited about stuff. They're working with us on two components in particular. One is the read-level data store, for which we're now piloting what is basically a custom implementation of Bigtable. And we have a BigQuery-based variant store, which is actually quite whizzy, but not clearly, I'm not sure it's going to meet all of the needs. Again, very, very early. Genome Bridge today has a half dozen engineers of its own. It's not a big group. They're continuing to hire. We have another 8 or so engineers on this from the cancer genomics group and the production sequencing group and the genome sequence analysis group, so the 3 different major pipelines in our world. We inside Broad are starting to think about what this means for us. And not surprisingly, as Genome Bridge and we talk to a number of outside organizations, there's still a lot of discomfort with public cloud. This will change, but right now there isn't a lot of HIPAA data in the public clouds. There is such data there, and we in IT know that they do a much better job of security than we do. But from a perception standpoint, it feels to me like we're still about 3 to 5 years away from broad-scale comfort of putting clinical grade data in public cloud. So we're now starting to pilot out. Yep.
So they've typically not been willing to sign BAAs
Yep.
with institutions and take any liability. So is that changing?
Yeah, well, I mean, there are different ones. So Microsoft will absolutely sign a. Yeah, yeah.
Amazon and Google specifically, since that's what you've been working with.
Yep, Google is committing to do this, but Google's a little funny because they're not. We're finding it challenging to work in the Google cloud because it's a little less mature, obviously, than the Amazon cloud. But I think one of the big differences is that Amazon actually uses their own cloud for internal use, and Google doesn't yet. So there's not clearly a long-term commitment yet to the Google Cloud services. Amazon has signed only a single BAA that I know of, and that was with DNA Nexus. But they did sign one.
So there's hope.
Yeah, so there's hope. So what we're doing now is we're piloting an internal cloud. We are now building out an OpenStack-based cloud infrastructure that the Genome Bridge development team will start porting their work towards. And we will see what we need to do over the next handful of years if we actually have to build out one more generation of infrastructure to host this, to drive both Genome Bridge and BRIITE for the next 5 years, or whether we can move more aggressively to public cloud services. It's still the case that I can do it much more cost effectively than the public clouds, but this question of elasticity at scale becomes huge. So we're all waiting now for the build 38 of the human genome to be released, and all the researchers will want to go and redo everything that we've already done. And that will take us a substantial amount of time in-house, regardless really of the scale of our infrastructure. We need something quite big. So anyway, so that's all I wanted to share about Genome Bridge. I'm happy to take questions.